So moving on to the section on fertilization. So we've looked at the menstrual cycle of a non-pregnant female. Now we're going to look at what happens if conception occurs. The egg has two layers, and both of these layers must be penetrated by a sperm before the egg is fertilized, or the ovum. Again, we call it an ovum typically as a human. There's receptors on the ovum membrane that sperm will attach to. So little receptors, not a lot different than receptors that we draw, but there you've got sperm. I can even draw the mid piece in there if I can get it. There's a sperm, the mid piece, the tail, and there's going to be hundreds of sperm that make it to the egg. And it actually takes hundreds of sperm to have enough acrosome to digest through those areas, and then one of those sperm will get in. You don't have to worry too much about a lot of the names here, but it does describe the names of the structures around the egg. The first polar cell I'll refer to in the next unit. The nucleus is important, and the idea that it's a haploid nucleus, which means that it just has half the chromosomes for a normal human complement, and then what we would get is the other half from the sperm. Okay, so typically there's 46 chromosomes. There's some variation in that, but typically 46 chromosomes. The number of chromosomes in the egg is 23. The number in the sperm would also be 23. We don't want to forget that there's other organelles inside here that we don't see. They're not being drawn, but they are there. We're just taking them out just so that it's easier. So we've got endoplasmic reticulum, mitochondria, Golgi apparatus, vesicles, ribosomes, and so on inside the cell. And when we look at even just the proportionate size, this diameter of the egg cell is 110 micrometers. The sperm are significantly smaller than that. They only have a nucleus and that acrosome in them that's going to help digest through these layers here. So they're not carrying any organelles with them. So the organelles that are contributed to the zygote are going to be only the organelles from the mother. So once the sperm have attached, the receptors will change shape. That discourages other sperm from attaching. The acrosome is going to release enzymes, and you're going to get a whole bunch, or going to require a whole bunch of enzymes that are going to be needed in order for that one sperm to get inside. And when that one sperm gets inside, there's a bit of an action potential. It's the best way to describe it, that is produced in the ovum that causes a bit of a surge and makes the egg swell a little bit, and it pushes all the other sperm off. We only want one sperm fertilizing the egg because we need 23 chromosomes plus 23. Any more than that, and you're going to get a zygote that isn't going to live for very long. It's not very likely that it's going to live very long. So when we do get that fertilized egg, we now can call that fertilized egg a zygote. So we can see where the zygote is on this diagram down below. We've got it right here. I'm just highlighted in yellow. Fertilization needs to take place in this first third. So the sperm has to enter up here. And the reason for it is because there needs to be a number of cell divisions that happen for that zygote to develop the structures that are required for implantation. So it needs about 6 days. So the zygote moves down the fallopian tubes towards the uterus, dividing along the way. So we're going to get 2 cells, 4 cells, 8 cells, and you can see that called cleavage here. Cleavage just means the splitting. And so we have the same sort of overall size here, and then it splits into 2. We got the same size of area, but it splits into 4. The cells get smaller, but the actual sphere of that now was a dividing zygote is not getting any smaller. We call it around this stage here a morula. So we'll go back up to the notes here and highlight that word. It's an 8-celled structure in Bio-30. Arguably, it could be up to a 32-cell structure. But the idea is that this morula is a solid ball of cells, and they are undifferentiated cells. And that means that they haven't decided what they're going to become yet. They're essentially stem cells. Now, identical twins can occur at this point, where you might get a couple cells from this morula break off and start dividing and dividing on their own, while that original ball of cells continues to divide as well. If they both divide to the point where they can develop the structures that can implant into the endometrium, you could get 2 identical twins that come out of this. Now, they're going to be identical because they came from the same DNA, right? It was just like a break off from that original cell ball. But if you have fraternal twins, you'd actually have to have two eggs fertilized and be fertilized by two different sperm. So we'll talk a little bit more about twins in the next unit. So as it progresses through the fallopian tube, going through these divisions, it will end up forming a structure called a blastocyst. And a blastocyst is a hollow ball of cells. So I think of a morula like a little chocolate ball that has bubbles inside, like the arrow spheres that you can buy now, not the arrow bar, but the spheres. And if you bite into it, you can see all the bubbles, but it's a solid ball of cells. If you think about a blastocyst is a hollow ball of cells, something like a Kinder Surprise, where you have like a chocolate layer, and then there's space in the middle, and then think of that little toy on one side. There's a toy on one side, and that toy would be the embryo, if we're using that as an analogy. So the outer chocolate layer of cells would be the chorion, and then the embryo would be that little Kinder Surprise, as well as any of the what we call extraembryonic structures that are associated with the embryo. So things like the amnion and so on. So then there's that hollow space as well inside that blastocyst. When we look at a blastocyst, so if we just look down on the side here, I'll maybe use a green so you can see it. This is your blastocyst. What it'll start to do is that outer sort of chorion of cells here will start to grow villi, or little finger-like projections off of it to try to, and I'm going to move into the uterus here, implant itself into the uterine wall through the endometrium. And by growing those fingers, it's sort of like rooting itself into the endometrium, which is going to allow for an area of diffusion of oxygen, wastes, nutrients, as well as create an attachment point for the long-term plan that this blastocyst has to hang out in the uterus for 9 months. The blastocyst also is going to end up forming at that area eventually a placenta, which will develop around the 3-month mark, because this embryo in this blastocyst is going to keep getting bigger and have more demand for nutrients and oxygen. It's going to need something bigger to be able to support it. So the other piece that's on the next page, but I'll just leave this diagram up for now, is this chorion here. It secretes a hormone, and that hormone is called human chorionic gonadotropin. And that is the hormone that is detected in pregnancy tests. It is the hormone that tells the body that the body is pregnant. So the only reason HCG should be in a person's body is if there's a developing embryo in there. And so it acts a little bit similar to LH, and it'll affect the corpus luteum, allowing the corpus luteum to stay intact a little bit longer. Well, it's going to be quite a long time. And it will continue to secrete estrogen and progesterone, which will continue to keep the uterus maintained by keeping it vascular and preventing uterine contractions. Once the placenta forms, it'll become its own gland that'll support the estrogen and progesterone secretions, but the corpus luteum will not break down, at least until that placenta forms. So just on the next page, I summarize that. So when fertilization has taken place, then once that chorion forms, the HCG is present. HCG is detected in pregnancy tests. So that'll be, when it gets released in the uterus, it's going to go into the bloodstream, right? Because it's going to have to communicate with the brain somehow. And then from the bloodstream, we know the blood gets cleaned at the kidneys. And so the kidneys will excrete some of that HCG, which then gets detected in a pregnancy test.